All right. A very good evening to our esteemed guests today and to our audiences joining us from across the globe. I'm Malvika, the moderator for today's conversation. Thank you so much for each and every one of you for joining us live today. This is the 7th episode of Biotalknology. And as you already know, Biotalknology is an initiative by Premas Biotech to create a platform for conversations with experts from the biotech industry. Our guests share with us their thought leadership, their views on how the industry has evolved, their journeys, and their perspectives on emerging trends and upcoming challenges. Today's topic of conversation is global challenges in animal health. And on the panel today, we welcome Dr. Mahesh Kumar, who is Senior Vice President, Research and Development at Zoetis. Dr. Kumar is a global leader in animal health and vaccine and has made significant contributions in this domain. From Premas Biotech, we have on the panel Dr. Prabudha Kundu, the managing partner and co-founder at Premas Biotech. The format of the conversation remains the same as before. After the panel discussion, the session will be open for about 20 minutes for taking questions from the audience members. I request the audience to kindly drop in your questions in the chat box, and as many as possible in the time frame that we have will be taken up and shared with the panel. Now, I don't want to keep you waiting. I request Dr. Kundu to share opening remarks and to welcome our guest speaker for the day.
Thank you so much, Malvika. Very warm welcome to all our audience, and especially to Dr. Mahesh Kumar. A very warm welcome to you, sir. I take this opportunity to do a brief introduction, and then we will get into our 7th episode of Biotechnology. Dr. Mahesh Kumar received his Bachelor of Science degree in biochemistry from India. He moved to the United States to pursue his post-graduation education in microbiology. He obtained his Master's of Science degree from the University of Maine and his PhD from the University of Minnesota in 1990. After graduation, Dr. Kumar was hired by the Maine Biological Laboratories in R&D. During his tenure at Maine Biological Labs, he developed several key products, including live and inactivated salmonella vaccines and bursal tissue origin line of infectious bursal disease virus containing vaccines, amongst others. After 7 years at MBL, where he was the head of R&D and regulatory affairs, he moved to Fort Dodge Animal Health. At Fort Dodge, he led the global poultry research and development activities, licensing several key products for the food safety, hatchery, and breeder segments. Notable vaccines developed by Dr. Kumar are E. coli and Avian Influenza, including a global approval for the use against the Asian H5N1 strain. He has several publications in avian health and is an inventor or co-inventor on several patents. In 2007, he moved to the Netherlands to head the regional technical office responsible for EU AFME approvals of the pharmaceutical and biological products for all species. He was also part of the regional management team providing guidance and diligence on all technical R&D issues. After the merger with Pfizer, Dr. Kumar was appointed to lead a new line dedicated to biological research in animal health based in Kalamazoo, Michigan. In 2013, Pfizer spun off the animal health division as Zoetis, where he currently serves as the Senior Vice President for Global Biologics R&D, responsible for vaccines portfolio in all species, including vaccines against transboundary and emerging infectious diseases. Mahesh, a very, very warm welcome from all of us.
Good evening and good morning or good afternoon, wherever you are. And thank you, Prabudha and Malvika, for the introduction. It is my great honor and privilege to present to you my thoughts on animal health and contributions to science. Once again, I hope all of you are well in this pandemic, and I wish you are keeping safe and dealing with it in an appropriate way. As we know, vaccines are going to become available soon, and we'll talk a little bit about that. And I'm so fortunate to be in this space for so many years and working on vaccines. So I'm very delighted to be here. And thank you once again.
Thank you so much. We're really fortunate to have you. Now, we will spread our discussion over 4 segments. And let's get to some of our initial thoughts on how you have come to where you are. You've had a very interesting career, Mahesh. And if you could start that, how did you get interested in animal health, and who and how did the last few years pan out? It's been very exciting, I hear.
Well, you're talking about my whole career in a nutshell, but essentially, I finished my biochemistry, and I really wanted to go and pursue a postgraduate education. It just so happened the University of Maine is a sea grant university, and I don't know if you're familiar with that, but in the United States, a university is a land grant, a sea grant, depending on the work on aquaculture-based medicines or products, or agriculture-based. So there was some federal funding for a sea grant fellowship, and I basically took that. And I was able to pursue a program studying fish diseases in Atlantic salmon. And so that was my introduction into animal health. I was not particularly looking for that space, but it just so happened that I fell into it. And then I went to Minnesota also on a fellowship from the federal government to pursue a sea grant fellowship, pursue my PhD also in fish disease. That's kind of how I went into it. And then as soon as I finished, I was offered this position at Maine Biological Laboratories, which happened to be a poultry vaccine company. And interestingly, they were really one of the pioneers in those days, and they essentially helped a couple of companies in India get started as well in the poultry health industry.
Thank you so much. Very, very interesting, because it's interesting, especially to some of the young scientists on the webinar today, in terms of how people get into such careers, because they would be a great interest. And maybe we'll cover a few of this as we go along. I think it's important at this time to also understand the contribution of Zoetis to global animal health. And if I have your permission, would it be okay to sort of share a few slides at this point of time? So I'm just going to, for the sake of our audience, just share some slides which I bring up for you. Are the slides visible to you, Mahesh?
Yeah.
Please, over to you, sir.
Can you make it full screen, or?
Yes, it's come up.
It is okay. All right. So as you know, Zoetis was spun off from Pfizer in 2013, and we became a basically completely independent company. And but just from a historical standpoint, Pfizer's been in the animal health industry for over 50 years. So I mean, from the standpoint of the introduction of terramycin, which is still being sold today, animal health has been part of Pfizer for some time. But when they spun us off, we became totally independent, and now we are the largest animal health company in the world, with the last year's sales up to $6.3 billion. Our focus is approximately 50-50 companion animal and livestock. Livestock comprises your cattle, sheep, goats, chickens, pigs, and fish. And our companion animal sector is basically cats, dogs, and horses. So between the two, we have a 50-50 split. We also have, I would say, a 50-50 split in the regional segment, the international and American. So this actually gives a wide view of things, and also allows us to tackle any difficulties we have in one segment or one part of the world by other parts of the world helping out. We have basically into all core species of animals. We have 27 manufacturing sites. We have over 1,000 R&D colleagues, and we are into 7 major product categories. We provide medicines, vaccines, diagnostics, genetic tests, biodevices, and now precision livestock farming, more digital and analytics that help the farmer pick up the best candidates for their farming. We have approximately 10,600 colleagues worldwide, and over 100 countries. I will stop right there from the introduction of Zoetis. I will just say that we are focused in a continuum of care within Zoetis, where we are, I would say, the 4 pillars for us are predict, prevent, detect, and treat. So we want to predict what might happen in the space and animals, and be able to prevent those diseases. We also want to have the proper diagnostics and genetic tests and tools so we can detect what's happening out there. And of course, if it's needed, provide treatment in the end. So we are basically in all spaces in animal health. I would say the only place that we're not currently is in pet food, but otherwise we are in pretty much all the other segments. Some of the important products we approved is Simparica Trio, which is a three-way combination product for heartworm, fleas, and ticks in companion animals. A Procerta line, which is a de novo vaccine of our Marek's disease and bursal disease and Newcastle disease. ProHeart, which is a heartworm preventative, which is a injectable 12-month product. Revolution Plus, which is another one for cats, which is also a spot on for flea and tick. Besides that, we also have many collaboration alliances. We started an innovation hub at the Colorado State University, building one at Texas A&M. We have a collaboration with Fish Vet Group. We also bought Platinum Plus, which is a equine health provider, and many other ones. So we are very proud to be a partner to many companies around the world, and we are happy to be leading in this space and continue to hope to deliver innovations in this space. We're also happy to work with Premas in some form or the other over the years.
Thank you so much. Oh, that's wonderful. Thank you, Mahesh. It was lovely. So coming to a very interesting question is, it would be very interesting for us to just sort of understand from your perspective that the issues in animal health that were prevalent when you joined the sector a couple of years ago, or maybe a couple of decades ago in that sense, and to sort of give them a view as, have we been able to really overcome some of those issues that were facing the sector when you joined versus now?
I would say in animal health, most people probably don't recognize or realize that we've had many diseases in many species. And we've actually have successful products in all of those over the years. So in the old days, there used to be antibiotics, feed additives, chemicals. That moved to vaccines in the early 40s and 50s and continues to this day. So I would say that for the most part, if there was any disease we heard of or knew, we were able to quickly tackle that through vaccines and treatments. So me coming from the infectious disease standpoint and the vaccine space, I would say that, well, if you woke me up in the middle of the night and told me, Mahesh, we have a disease, what can you do for me? I can probably give you an answer and they'll probably work 80–90% of the time. Those few times where it doesn't work is when the bug, the pathogen doesn't grow very well, or we don't have a model to duplicate the disease in order to prove efficacy of a product. But over the years, I would say we've been very fortunate to have tackled many of the problems we faced. The issues that we've had in the recent past or since I joined have been more political and regulatory in nature, in the sense there are agents that are can't be crossed between borders, right? Avian influenza is one. We have a high pathogenic influenza virus, which normally you cannot take a product from one country to another. You have to register it there particularly. And so there are things there that the governments do as well, where they try to cull the population rather than vaccinate, right? So they don't want to create an endemic situation. So I would say with barring those new diseases like African swine fever, SARS-CoV-2, other things that come up very new, which takes some time for us to tackle, we have been able to tackle some of those things quickly. Where I would pivot to saying from before, what's changed here is really the technologies that we're using today. In the old days, we'd grow the virus or bacteria, we'd inactivate them, and then we'll adjuvant them and provide a vaccine. Today, we use more rational designs. We use in silico technologies to find out what is a protective agent or epitope. And we're able to then, express them separately in expression systems, or maybe vector them into virus or bacteria and then provide them. Another thing that I actually pioneered in those days was gene deletion vaccine. So basically, the first salmonella vaccine, live vaccine that I developed, was a gene deletion where it was an auxotrophic mutant. What that means is this bug will not replicate without providing some supplements. While we can provide that in media for it to grow, in the host, like in the chicken, the salmonella could not grow because technically vertebrates do not form aromatic amino acids. So this was a deletion of aromatic amino acid pathway, and this deletion pathway was used effectively, and we developed a vaccine. So some of the technologies we've employed today weren't available in the past, and more today, these technologies are going to help us and enable us to tackle problems in a quicker manner, and also in a platform type of way, so we don't have to develop things from scratch anymore.
Right. Thank you, Mahesh. The one question which I had, and it was very interesting, and I think it's been a topic that you and I have spoken earlier as, but we are very interest to our audience is, why are there more animal vaccines than human vaccines?
That's a very interesting question, right? So number one, there's of course many more species of animals than humans. And also from the I guess the introduction of antibiotics, most of the bacterial diseases in those days was basically tackled through antibiotics. And really the research and development of vaccines for bacterial antigens or bacterial pathogens stopped some years ago. So when the advent of measles, mumps, and rubella, polio, diphtheria, pertussis, and tetanus, DPT, really that essentially stopped the vaccines from development until very recently with Ebola, chikungunya, SARS, MERS. Things have started picking up again, dengue. So there's a lot more interest today. But even recently, even the Prevnar, the pneumococcal conjugate vaccines was really a recent introduction for ear infections in children and other issues for older adults. So there's kind of a renaissance, if you want to call it, of vaccines in human health. But in animal health, that's not the case. While antibiotics can be expensive, plus because of the pressure on antibiotic use and the antimicrobial resistance that happens in human health, there has been a push away from using antibiotics. And we recognize early on the introduction of vaccines has essentially increased productivity and helped the production of these animals in intensive culture that really was a benefit to the producers. So in any livestock species, they look at every portion of production. And if you find that it saves money, they will embrace that. So vaccines was found to be very helpful in those days for that. Moving to companion animals again, it was quite quickly embraced, and many diseases out there that would say that were common to animals were done. The other piece of that is, of course, being in animal health, we're able to test these products in animals and then quickly ascertain whether it works or doesn't work. In human health, the cost of developing a vaccine is very, very expensive. You're talking billions of dollars. And the amount of investment needed if it's really not bringing it to the forefront and they don't see a clear return on that investment, people didn't go there. So very few companies continue to stick with it and continue to work on that. But if you look at the number of products, they have not been as many as in animal health.
Well, thank you. Absolutely. I think the ease to market or the time lag, and we'll cover this in our subsequent one, that what happened with SARS-CoV-2? How did suddenly the world wake up to this, rapid deployment of various technologies? I think you make some very, very valid points. Now, moving gears a little, Mahesh, coming to your personal experience on these three very important animal health vaccines, avian influenza, E. coli, and salmonella, that you and your team, and I know for sure that you were very, very, acutely involved in the design and deployment, the development of this. Could you take us, and it will be very interesting I was looking at some of the people in the audience, and there are some young scientists out there who are also involved in animal health, that when you did this development of these vaccines, what were some of the challenges and what were the things that you went through? I mean, if you could cover in some of these points briefly, like avian, E. coli, salmonella.
So I think the first thing is, if you recall, I said that these were novel technologies, right? Using gene deletion as an auxotrophic mutation into a bacteria so that it will not be able to replicate in the host, but only to limited ability and then protect that host was a novel concept. This technology for aromatic amino acid disturbance was developed by Bruce Stalker at Stanford University. This was around for many years, but Wyeth, our parent company, had licensed it. They actually worked on it for human health, human salmonella vaccines, but they abandoned it because of the cost. And then I picked that up from an animal health standpoint and approached that. I used the same arrow-A technology for E. coli as well. So two things here. In the old days, people were controlling E. coli. I will stop at salmonella because I've talked about that already. I'll just talk about E. coli a little bit. People were tackling E. coli in the poultry industry through antibiotics. And to this day, too, in many of the developing countries, they still tackle it with antibiotics. So here I was trying to show using an aromatic amino acid in a vaccine, E. coli vaccines could help the poultry industry. But there were not too many takers at the time, including our own commercial people in my own company did not believe this would succeed. Why? Because people thought E. coli was a secondary or tertiary pathogen. But actually in avian, there are three things that causes respiratory diseases and production deficiency. One is mycoplasma, the other one is infectious bronchitis, and the third is E. coli. And any one of these agents can be a primary, secondary, or tertiary pathogen. We already had vaccines for bronchitis. We had vaccines for mycoplasma, but we didn't have for E. coli. People were treating with antibiotics. So I made the case that having a vaccine would be helpful. Coincidentally, of course, many of the developing countries started a push towards no antibiotics ever in the poultry industry. Today, many of the big players like Chick-fil-A, McDonald's, Walmarts do not buy antibiotic-used chickens. So there's a lot of pressure on the government. So I foresaw this many years ago, and we decided to use the same arrow-A construct in E. coli. It's like it's an analogy in America about Field of Dreams movie where they built a baseball field in the cornfields, said if we build it, they will come, and then finally everybody came to play baseball in the cornfield. So this is a way we decided to build this concept and say, okay, somebody will want this. And today, I would tell you it is the biggest selling vaccine for a company in avian, which to me was a surprise because at that time, nobody believed, including our commercial people, did not want this vaccine. The other challenge is regulatory. Since it is a gene deletion, right, you have to use genetic means to remove that piece. So this has been difficult for some countries to accept, while we all know in the scientific sense, it is actually much more safer. And with these defined deletions that we take a large gene out. The chance of it having mutations or having it homologous recombined, come up with a more pathogenic product is not possible. So the education aspect and the convincing of the authorities is significant. So we have been trying for many years in many countries, including India actually, to try to get the E. coli product approved. And we have to having to go through many of the committees to convince them that this actually is a safer product than others. So we've faced challenges similar to that in many countries. Even in the EU, I had a catch-22 where they said, it's gene modified so you can't do field safety studies in livestock. I said, if I can't do the field safety study, I can't get the product approved. So what do I do? So finally, they accepted that we can do it in Morocco. And France accepted to use that data for the approval. Morocco was a French colony at one point, and maybe they took kindly to that. But either way, they took that data and got the product approved, and we got the product approved finally in the EU, and we have it successfully approved. And we do have it approved in many of the countries, but it's still a challenge. I'm trying to get it approved in China. I'm facing the same difficulties over the regulatory pathways and trying to understand what it would take for us to convince the people that it is safe and efficacious and that it is truly a benefit. Let's pivot to avian influenza. This again was a first in class, right? You're talking about a reverse genetic system where we have 8 genes that make up avian influenza, not just avian, any influenza. So we took the backbone of the H1N1 flu virus from humans, which is a it's called a PR H1N1 strain, which has been around for many years in the labs, very attenuated. So we used that backbone, and then we introduced a HA gene from the H5N1 Vietnam outbreak of the bird flu. And then we used the neuraminidase gene from a duck isolate from Germany. So it became an N3, not an N1. So that we could use a DIVA test to discriminate between the H1N1, I'm sorry, right, H5N1 and H5N3, which is a vaccine. But the governments could not understand that N3 was really a benefit. But they say, oh, you don't have H5N1, so we cannot license a product. You need H5N1. And I'm trying to explain to them, the N3 was made by us specifically so you can discriminate between the vaccinated chicken and a non-vaccinated chicken, which is naturally occurring. But this still, I'm still trying to convince some governments of this and still not being allowed, whereas they are using indigenously grown high pathogenic strains in vaccines. So using reverse genetics, I'm also able to remove a polybasic amino acid repeat in the HA gene that attenuates a high pathogenic virus. So by using it in plasmids, we're able to remove the repeat of the amino acids, which essentially attenuates the virus. So using genetic means it makes it much more safer and much more easier to develop these vaccines. In fact, recently there's another outbreak of H5N1 in the United States, which was a different variant of the H5N1 from Vietnam from 2001. So we quickly deployed the same technology and used the HA gene from the newest outbreak, removed the polybasic amino acids and inserted it. And we have a second generation bird flu vaccine, which we're using in some countries. So we are able to quickly move and quickly apply these technologies to deal with any new issue.
Well, thank you, Mahesh. That's really enlightening. One of the things which I picked up from your comment was that the E. coli vaccine actually helped you bring down microbial resistance. Is that what I heard, because of use of less antibiotics? Because that's a very, very important issue. One is to vaccinate and not use, but also bring down overall the antibiotic use.
So I actually, we did 2 studies to see the effect of vaccines on antibiotic use. The first one is a study done in Norway in salmon industry, right? So you think about the tons of antibiotic used in Atlantic salmon, and Norway being the world's largest salmon producer, and there were several 100,000 tons of antibiotics being used in there. And then since the introduction of first vaccines in the early 80s, I can show you a graph where the use of vaccines had steadily increased and the use of antibiotics has steadily come down, where today the tonnage of salmon produced is like 100 times more. The antibiotic use like 1,000th what they were using. So we clearly could see a trend of vaccine use and the commensurate reduction of antibiotics. I can show you today also same in E. coli and the poultry industry, where you can see clearly the impact of vaccines and the use of antibiotics. We can see, and of course, the vaccine of the E. coli vaccine has only been recently introduced in the last 5 years in many countries. So data is still being generated, but we have clear, significant data we have shown where vaccines have used has been a significant reduction of antibiotics. We also know the intensity of the antibiotic use also reduced, meaning if we have vaccines, the number of days they had to treat with antibiotics dropped from, say, 10 days to maybe 2 days. So not only was the sheer amount of antibiotics reduced, but the number of days the antibiotics used was also reduced. We have data to show that as well.
Fantastic. That's really, really impressive. Now moving gears again to our third topic that we have here and looking at is the pandemic, SARS-CoV-2, and how the world has sort of reacted to it. And before that, would this be the right time to share with the audience some of the other slides that you had? And I think it's very important that we get there, if that's okay, Mahesh. So
Please.
So this is what we have. The animal health contributes to our health. And over to you, Mahesh.
Yeah, if you want to look at the slide here, this actually is Edward Jenner, who I consider the father of vaccinology, inoculating this 8-year-old kid with cowpox. And these were days before a lot of regulations. So he basically, well, you know the story that at the time of smallpox, which was rampant around the world in the 1700s, he found that this group of milkmaids were not getting smallpox, but he noticed that these milkmaids in the hands had these small pustules that resembled smallpox, but they were not smallpox. And he found out that actually was a related disease called cowpox. And these ladies who had this minor lesions of that were not getting any smallpox. So he thought, well, if I were to use this and try it, maybe the benefit would be seen. So he got this young kid, and then he took this material from this milkmaid. He kind of macerated it, and then inoculated this child with the cowpox. And then what he did later was essentially gave this child the real smallpox, and he found that it protected him. So you can't do that these days, but he did do that and essentially fulfilled what we call Koch's postulates. So I have to say that animal health, even in those days, contributed to a significant portion of human health. And there's many, many examples of how One Health, or what we call One Health, has helped the industry and how it helps human health also. So I would say that if you look at the contributions that animal health has had, as I mentioned earlier, maybe move to the next slide, P.K. As I mentioned earlier, there have been many, many vaccines in the veterinary space that have helped protect animals from diseases. If you look here, this is really our Zoetis and our legacy companies' response to zoonoses. Zoonoses are an example of diseases that can actually move from animals to humans. There are many examples of those, Salmonella being one, you know that Avian Influenza is another one, and others that I have here. But basically, I have here a lot of emerging infectious diseases in the animal health space that we have developed over the years. If you looked at all the ones in the red circles, these are actually vaccines that we have made, but also sold to government agencies as vaccine banks. As you know, I told you that many governments do not vaccinate for high-path avian influenza. They prefer to cull the animals. However, they always want to have an insurance card. So what they do is they buy vaccines and stockpile them in case there's ever a need to vaccinate, then they're not scrambling to make vaccine. So we have supplied the US government, the EU government, the Dutch government, many governments around the world, vaccines that have been circled, West Nile, many influenza vaccines, swine influenza, equine hendra, which is a zoonotic pathogen and is related to Nipah virus for Australia that kills humans and horses, avian influenza H5N1. And so many, many of these have been stockpiles. I also have in rectangles the many, many coronaviruses we've developed over the years. So people believe that the SARS-CoV-2 is the first coronavirus, and man, how are we going to make so many vaccines for the world? We need billions of doses. Who's going to make them? Frankly, if I tell you that we've been making coronavirus vaccines since the early 1900s, you should not be surprised. The first coronavirus bronchitis vaccine was probably developed sometime in the early 1900s. However, we have I just showed you some examples recently of variant coronaviruses of bronchitis in poultry that we've developed. What I'm trying to say is there are a billion chickens on the planet on any given day. And these broiler birds that are meat birds turn over every 6 weeks, meaning there's a new set of 6-week-old eggs being hatched to be next set of broiler birds that will grow and marketed every 6 weeks. So you can imagine how many billions of birds are on the planet in a year. But on any given day, there's a billion chickens on the planet. And every one of these chickens get a bronchitis vaccine, which is a coronavirus vaccine. So making billions of doses of vaccine is nothing new for animal health. We have done this day in and day out. So all of you are worried about vaccines, rest assured, we will have vaccines for all of you when the time comes. And hopefully, as you know, we're very, very shortly going to get approvals. We already got the first one in the UK. I believe India is going to approve one soon. I believe this Friday, we're hoping the US will approve the vaccines in the US as well. So I want to tell you, we have made this many, many times. So how does this relate? How does this help us in the SARS-CoV-2 topic today? So what happened was in February of this year, when this pandemic was just beginning to spread in some parts of the world, we noticed that one dog, I think it was a German Shepherd Alsatian in Hong Kong, that was infected SARS-CoV-2. So we got immediately very interested, said, okay, wait a minute. If this is happening in dogs, we need to be prepared. So as the leading animal health company, it clearly would not be wise to sit this one out. And we said, okay, what can we do? So we immediately said, okay, hey, listen, we've made coronavirus vaccines all the time. So why couldn't we do the same thing for SARS-CoV-2? The problem here is the SARS-CoV-2 is highly pathogenic, and we can't bring it into a facility to grow this because, we would have to quarantine everybody, and there's lots of restrictions. So how do we prevent that from happening? How do we do this? This is where the newer technologies come in, what I told you earlier. How can we use the newer technologies to help us make these vaccines? So we go to the next slide, which you had briefly there, PK. So what we knew was in coronaviruses, they call it corona because it looks like a crown, and these spikes are around the virus. So in coronaviruses, we knew the spike, which looks like a trimer, which you can see on the left-hand side of this picture, like a rosebud with 3 pieces. The S1 trimer is known to be protective. We all know that. The problem is, up until the last couple of years, we were unable to express the spike trimer in its native form that would be stable, meaning you'd express it and it would basically become a monomer. It will not remain its trimer configuration. We also noticed, and some publications have said, that this spike trimer has to be in the pre-fusion state, meaning as it gets to a fusion state, it opens up, and the receptor binding domain, when it's open, is not as efficacious, meaning in the pre-fusion state, the antigen actually was 16 times more, had more neutralizing antibodies when you vaccinate with the post-fusion state. So it's very important for us to express this trimer in the pre-fusion state, as well as keep it in a trimer configuration. So what we did was we did a few things to keep it in a pre-fusion state, as well as in the stable configuration. We also had a Histax so we can purify the antigen, and we decided to express this in, we essentially transiently expressed HEC and then in CHO cells. And after we expressed this, and we did Western blots and did some work, and we were convinced, at least scientists convincing me, that, hey, we do have it expressed well, but I wanted to see it. I said, I know that, but I want to see that structure because I've heard so much about it. So we collaborated with this company in San Diego that was able to do a crystal structure of this. And you can see on the right-hand side, it's black and white crystal structure picture. But you can see this in a very nice trimer form. And you can see the S1 trimer and the ectodomain as well being expressed nicely. So, and if you can see this on the further right, I don't know if you can see it with my picture and PK's picture there, but we overlaid it with the crystal structure that was published, and it fit very, very nicely.
Yep.
And talking to those people, they said, we are also doing structures for many human health companies, and I have to tell you that your expression and the quality of this is so good that we want to see what we can do to help provide these human health companies as a positive control. So they were asking us to collaborate in this space. So our scientists, of course, very, very proud of that accomplishment. And then we were convinced this was good. And so then, of course, once we have the antigen, it's very easy to make a vaccine. Why? We have adjuvants and we have tested methods to make coronaviruses vaccines for all species. We have coronavirus vaccines for horses. We have it for cats and dogs. And we, of course, our primary goal here was knowing that the dogs were being affected. We wanted to make a vaccine. So we quickly formulated this with an existing adjuvant for companion animals. We also used a novel adjuvant because I wanted to actually enhance the T-cell immunity as well, not just the B-cell immunity. So we did 2 different adjuvants, one traditional and one novel for both cats and for dogs. And then we decided to vaccinate dogs and see what what antibody response we had. And as we were doing this and started working, this was in February of this year, and by March, April, we already had the vaccine developed. We decided, well, listen, if we were doing this, we need to have a test also in case this becomes big. How do we test our animals, right? So we developed a PCR, RT-PCR assay that was already being recommended by the CDC. Interestingly, the CDC's test was actually an older method to transport the virus, which was meant to keep the virus alive, so they can use it for isolating the virus. We actually said, you know, that's dangerous because we're looking at RNAs. Who cares if the virus is dead, right? So we actually changed the transport medium so they inactivate the virus. So it's safer for them to send samples to us in our reference lab. As soon as we developed it, we had validated, we immediately deployed it into our reference lab. We have many reference labs for animal health in the country. So it was already being offered. And if you see that slide, I'll show you next. The first dog that was identified as positive for SARS-CoV-2 in the United States was in our reference labs.
Oh.
So I'll get to that in just a second, but this is just a data set of the study that we did. So after we vaccinated, we gave 2 vaccines, day 0, and 3 weeks later, we gave another shot. We bled them at day 0, which was a an older dog. We gave it, and then 3 weeks later, another vaccine, second dose, and then we bled them at the time of vaccination, first time, the time of revaccination, and 3 weeks after that, we still continue to monitor them. If you look at the top part, you see the T1, which is all control animals. You can see the serum neutralization assay, they're all negative. You can see the lateral flow, which is just basically like a pregnancy dipstick type test. And then you can see on the way right, we also developed an ELISA. You can see clearly the controls were relatively neutral, meaning negative. You see the second group here, T2, which is really our conventional adjuvant that we use. And the third one is a novel adjuvant. You can clearly see both the adjuvant groups with a spike trimer gave excellent responses. What was more interesting was the novel adjuvant really kicked it up even more. And we continue to see that. And I have to tell you that it's now 8 months, 6 to 8 months. We're still seeing a good titer in those animals. So it bodes well for the vaccines you all will be facing as a group, right? So this is the canine study. Go to the next slide, I'll show you a feline study, which is in cats. And we did the same model. We gave 1 dose, a second dose 3 weeks later, and we bled them. Here again, you can see that the controls were all negative using the serum neutralization, lateral flow, and ELISA. And you can see that in the vaccinated group, they were actually quite good. Here you see that even though the novel adjuvant slightly better in one assay, in the ELISA, I don't see that in the lateral flow. But regardless, we have a good adjuvant system for this. So we feel very comfortable that we have a great antigen. The upstart of this is the virus is the same, right? Whether it's human, for cats, dogs, the virus is the same. So technically, we actually have a very good antigen. And actually, we are looking for partners in the human health space. If they want it, we can actually supply them the antigen. And then there's plenty of adjuvants they can use and harness from the existing adjuvants in the human health space. But again, I think if you wanted to have an antigen, I'm very, very happy with what we have. As an aside here, it's actually been published in one of the congresses recently. We also tested the AstraZeneca or Oxford University's adenovirus vectored feline, adenovirus vectored chimpanzee vectored vaccine for SARS-CoV-2, and we tested in the cat along with our adjuvant. And I say that their vaccine works. However, I would say that our adjuvanted one was much, much better in titer and response. Now, I can't say the vaccine was worse because that was meant for humans. We just used the same construct in cats, so maybe it was not optimized. So I'll give them the benefit of that doubt, but it clearly works in our model. So what we did was, as soon as we knew this was working, and we said, well, we also want to make sure, like I said, an antigen test as well for the cats and dogs. So we developed this test, the PCR test for animals. We also developed a lateral flow and ELISA test for animals. But then, as I told you, the antigen is the same. The antibody would be the same. So what would it take to develop it for us, humans? So we basically put some controls and found it worked. And actually, to be honest with you, we are offering it as a test for our colleagues here in our company for our own people, and we are applying through the FDA for a clear waiver, so we can use that for a broader audience. So we're hoping to offer it for Kalamazoo in our company and the Western Michigan University's medical school for our people. So it's a win-win for us. And I just want to show you a slide or two on the assay for antibodies and the diagnostic space. So if you go to the next slide.
Absolutely.
I'll
Mahesh,
show
as we
you.
go, I just want to also make, I mean, I don't want to stop you, but there are a lot of questions coming in, I believe. And so if it's, I know,
Okay.
so we have maybe a couple of minutes.
That's fine.
No problem, please. Just go ahead, please. No, just want to do, yeah, please.
I think we're almost done with this. Just wanted to share with you very graphic. I don't need to explain it, but everybody here probably knows RT-PCR. We do the same nasopharyngeal swab. We collect it, transport medium, extract it, and we get a PCR result. So next slide. And if you look at the lateral flow again, if you can see that it's really very simple. We are able to take, this is just a graphic to show the dogs that were tested positive by us in our test. This is a lateral flow assay. Essentially, you can take a piece of the blood, a drop of blood, drop it into the well. You drop some chase buffer. You wait 10 minutes. And if you have two lines, it's positive. So one line is negative. So it's very, very simple. It's actually in 10 minutes you get the stuff. And like I said, we have also done it for us. We take a finger stick, we have a little pipette, which takes only 25 microliters. You drop it and you have a positive or not. 10 minutes, we have an answer. So we're very, very proud to say, as a Zoetis company, we were able to really move when we needed to, and we're very dynamic in what we could do. And I have to thank the company for allowing me the freedom to really go after something that's not even an official project, right? So if something happens, we're able to delve right into it and really pivot to what we need to get this for not only the animals, but actually for our own colleagues as well. So I want to stop right there, and I have to say that once again, I want to thank all of you for your attention and the opportunity for me to show off what I really do. And thank you, PK and Malvika, for this opportunity, and Premas as well. So once again, I thank you very much.
And Mahesh, before we go on to the question answers, there's something that I want to personally, ask you, and I think it's going to be, again, very, very important for our audience. So if I can kick that off is, what do you believe? There are two questions, actually. What is the future of animal health weigh in for human health as well? That, and what are the top 3 lessons that you've learned on the way up till here?
Okay. Well, the future of animal health is very bright. I think people consider the companion animals as more than human. The animal-human bond is increasing. People probably, I can tell you personally, my wife takes care of my dog better than she takes care of me. So the animal and human bond is significant, and that's going to only grow. From a livestock standpoint, we know the amount of arable land is getting more and more less. And so we need to be more efficient. So many of these products come into play to help increase the productivity. So I know for a fact that the animal health industry is going to be continuing to grow, and it's going to be something that's very much needed, not only from the animal health standpoint, but also as an exemplar or a way to show what can help us in human health, because we can do things more quickly in animal health. Of it, right? The other piece really is that every pathogen, just like any of us, wants to survive. So every time you have solved an issue, guaranteed there will be another pathogen waiting in the wings to take its place. So I can tell you that I will always have a job, you will always have a job, and all of us in animal health will always have a job when it comes to pathogens and disease prevention. So we will have to be really watchful and vigilant to make sure we're able to take care of the new issues that come up. And as far as the lessons, I would say I was actually giving the same thing to my own son, who just graduated from his college and is moving into the world. I told him, what people look for, I look for in employing somebody is the right attitude, right? I can have the smartest person on my team, but he has a very negative, pessimistic attitude. he doesn't belong in R&D, right? Think about R&D. It's research, research. You're going to fail 90% of the time, right? So how do you keep motivated? How do you keep positive? How do you keep going that one next step, one next study to do that? So I think you need to have a positive attitude. I want people, so whatever you are, wherever you are, whatever career you're in, I want you to have the right attitude. That's one. Two, be sincere, right? I mean, we're all here for a short period of time. There's no time for you to try to be one up on one other person, right? Do not look good at the expense of someone else. I hate that. I tell my people, right? I give you all the respect and trust, but the minute you try to look good by putting somebody down, you are at the bottom of my trust ladder. So be sincere and be authentic, right? Don't need to be somebody you're not. Be yourself, and that's going to take you a long way, no matter where you go. So those are two lessons. The third one is a golf analogy that I like to golf quite a bit, and the PK knows, and some of who know me knows this. Is I think several people said this, but the most famous guy says Gary Player. He said, "Man, the more I practice, the luckier I get." Okay, what that I take to all of you is that working hard is a given in this business, right? You come in and that's something that's a given. I don't expect people to say, "Oh man, I don't want to do this," but you need to really put in the time. You need to work hard. The rest will come easy if you work hard. So like I said, if you keep practicing a lot, you work hard, luck will come. So you'll be lucky. That's all I will say. And again, thank you again for this opportunity and for all of you for listening. And I'm happy to address any questions you might have.
Mahesh, can't say how blessed at least I am, and I'm sure our audience feels the same. So thank you so much, really. Malvika, let's get some questions going. I think there are quite a few questions in the wings.
Yes, absolutely. Thank you, Dr. Mahesh and Dr. Prabha, for this amazingly insightful session. We do have a lot of questions flowing in. The first question is actually related to whole supply chain. Professor Gautam Ghosh asks, for developing world animal healthcare vaccine has different challenges, such as administrating it in far-reaching areas where maintaining cold chain is often not possible. Do you work on developing thermostable animal vaccines as well?
Yes, I do. And this is a question that comes up all the time. We have projects with the Bill and Melinda Gates Foundation as well, trying to help on this space. And we've been working on this. The problem that I have found is, how do you define thermostable, right? If you ask 10 people what thermostable is, they'll give you 10 different answers. So one, if I ask somebody, thermostable means that it can stay alive and efficacious sitting at 80 degrees Celsius for 10 days, okay? Well, that's impossible to do that. So I think defining a practical thermostable parameter will help in success. Meaning, if you have a vaccine that's out of the cold chain for maybe a few hours, even at a higher temperature, is that good enough? Then you can have the last mile, as they say, parts of things that can be done. So you don't need to have it in a refrigerator, maybe some ice. Even if it's warm to a certain degree, it's okay. So I think a practical definition of thermostable is very important when you tackle these subjects. I think many academics too get waylaid and then distracted by going after this true thermostable definition and spend a lot of time and effort and never get anywhere, because you will spend a lot of time to a point where the return on that investment, there's not much, you're not going to get there. So I would say define what is thermostable to you, and then figure out what the best way to tackle that is. And then there are ways to do it, but you need to be clear on what that is. You're having two problems. You want thermostable and cost, right? Whatever thermostable technology you're going to develop has to also be cost effective for developing countries.
In fact, Dr. Rajkishore Gogoi also asked if there are any other development in this area for making sure that there's no vaccine failure while sending out these vaccines to such areas.
Well, I mean, there are things that we are trying to do. I'll tell you just a very simple story, and this goes back to ancient times, right? there's this seed in the desert, right? And then it's a desert, there's no rain or nothing. But the minute there's a drop of rain, right, it'll bloom, right? So people found what's going on, why does it happen, right? So basically they found that this seed is protected by sugars. And as sugar is a very good cryoprotectant, protects very well. So so as soon as the water is in there, it survives and goes back. So question is, so if we can protect something in the desert heat for hundreds of years, and just a minute you drop in water, something happens, how come? Why can't we do this to bugs, right? So the trick is this: to stabilize something, you need two things: sugar and protein, right? Sugar is great in one sense, but the other part of sugar is also traps moisture. So you have more moisture, it degrades the antigen. So how do you balance the amount of sugar that gives a great cryoprotection along with the desiccant, like a protein that keeps that? So doing the balance is the trick here. So people have tried many different parameters to do this, trying to get into a glass state. So once a glass state, you can powder it and do that. So there are techniques that you can use, different, but it's really a trick of a sugar and a protein. So you, as a scientist, you all can figure out what to mess around with. There's a whole toolbox out there. You kind of figure it out, do your diligence, check it out, and then try different things. And I would say you may be successful. And if you know it, call me. I'll license it from you.
Thank you. Sankar Bhattacharya asks, are there any regulatory blocks for live attenuated virus vaccines for farm animals?
Yeah, I mean, different countries have different regulations. The easiest path is if you have a live attenuated vaccine that's naturally attenuated. The way you do that is you basically passage that strain over and over. So you grow it multiple times. Maybe for coronavirus, for example, they will attenuate between 80 to 100 passes in a non-traditional media, meaning if the virus grows in chicken cells, use duck cells. If the virus grows in Vero cells, use some other cell. So by doing that and passaging 100 times, you'll attenuate it. Then you have to test it to see safety versus efficacy. Sometimes too attenuated, it won't work. However, when you look at an attenuated, live attenuated vaccine, the governments want local strains, right? I can't bring something from Africa into India or India into China because they will say, wait a minute, you're bringing something foreign to this country. So yes, live vaccines are allowed. You'll need to prove that that's safe and efficacious and that's indigenous to that region, whatever that region is. You have to go through more hoops. You want to bring something different by showing that the same virus exists in that region, meaning the genetic means you can show phylogenetically by sequencing that they are the same or not. That's another way to do it. When you do genetic deletions, like gene modified, even those techniques are all available today. You can easily do that. It's very safe. The regulations are higher, the barriers are higher because of the whole GMO cloud. And so you need to convince the government agencies that indeed you can do this and it's okay. And there's a little more, they can be done, but it takes a lot more effort. And sometimes it takes many years to get there.
Got it. Radha Chauhan asks, what's the criteria for accepting a candidate as a vaccine in the animal world in terms of the efficiency?
That's a good question. Actually, that depends on the product and if it's already published, meaning there are pharmacopoeia, or in the US it's called the Federal Register, publication already published parameters for certain pathogens. So if you want to make a vaccine for that disease, it's already laid out as to here's what you need to do. But in general, 80% take in the controls, meaning 80% of the unvaccinated animals should come down with a disease or clinical symptoms, and then 80% of the vaccinated animals have to be protected. There are also variations of that. We call the protective index. And there's also other mitigating factors you can show, say, hey, by these multiple factors through statistical means, I can show there's a benefit. But the hurdle or the bar becomes higher and higher when you try to squeeze things, tease things out over and above or less than the 80% threshold. So generally speaking, you have 80% threshold. If you have something that shows 80% of animals are sick and 80% of vaccinated are protected, you're good to go.
Got it.
Most countries.
All right. Patricia asked this question: what are the new animal health vaccines that the industry is working on now, and for which species?
Well, that's a loaded question. So some I can speak of, some I cannot, because protected or proprietary, right? So I can tell you some of the recent ones we've developed. Okay, so I already told you the SARS-CoV-2 is one we're working on today. So obviously, we are trying to get this approval. As you know, the USDA has still not acknowledged that it's a significant disease in cats and dogs. Of course, we can't be waiting for them to decide it's okay, because then it'll be too late. So we are working towards that. But we do know that minks are being affected significantly around the world. Close to 100 million minks have been depopulated already in Denmark and Netherlands and the US. So there is a market for the USDA recently said they will consider vaccines for the mink. So we already have data. I just shared with you the data on the cat and dog vaccine. So we are showing that to them along with a plan to get a conditional license, or what you call emergency use in animals, to get a mink license. That would I would say the most current one that I'm working on that I'm willing to divulge, right? But if you think about other programs, you can probably guess some of the real other problems on pigs. It's African swine fever, which is a big deal around the world. It's destroyed multiple pig populations in China, which is a big source of protein for them. It's entering the wild boars in Europe, and it's causing a big problem. That's a big problem in pigs. Look at companion animals. You're looking at novel products out there from the standpoint of Lyme disease, other things that are out there trying to get in there to do better constructs out there. He's talking about canine influenza. There was a new strain of canine influenza that came from dogs itself. Originally, the first strain came from horses. That's something that we've done a vaccine for, to continue to do horses. If you look at chickens, of course, the in novo, meaning giving vaccines in the egg, is a way to go, and adding more antigens in novo is something that we're looking to develop and continue to provide vaccines for that. In fish, of course, already the salmon industry has already seen many products out there. The growth there is going to be the tilapia industry, which is very, very big. The problem with tilapia is they're so small. To vaccinate them at that young age is a difficult proposition. You're talking about how do you get a vaccine to a tiny fish. So there are many, many challenges there. They're looking at robotics technologies to do that. I would say another big thing that we're looking at is data and analytics. How is big data going to help us try to see what can happen? Just imagine that you have a range in Australia where the cattle are let out on the ranch. They don't see those cattle until a year later, meaning they vaccinate, only see them a year later. How do you know that animal is sick or not, right? Can you imagine maybe having a technology where you have collars that will communicate to drones that will tell you if that animal is sick, if it's sick, where actually the animal is. You don't know where those cows are. So how do we know where that is, who it belongs to? Maybe we'll tell them. Maybe you can tell, okay, that cow is sick. Maybe you find a drone to inject that cow. So technology is evolving, and many, many things are going to happen that we haven't even seen or envisaged. So it's an exciting time. And so, Patricia, I think there's many, many exciting things that we're looking at, working on. And I'm sure the animal health 10 years from now probably will not even be close to what it is today. In one sense, in the other sense, I always say, you don't have to do anything sophisticated. You don't need to do. Sometimes a simple, tried, true method that's effective products is good enough.
Right. This one question, and we're kind of like drawing towards the end, so I'm just going to like pick up last maybe two questions. One from Yale, who asks, is the lateral flow suitable for swine blood?
It should be. If it's able to test, it should be. The only thing I don't know and I can't remember is we have the right antibody control validated yet. But yes, the test itself would be suitable because it's essentially detecting the antibodies against the antigen. The antigen's what's dotted onto the membrane. But I have to. I don't know if I've tested for swine yet. But I think when we looked at the swine space, and I remember looking at the data was generated, I think we have information to show that swine are not as susceptible to SARS-CoV-2. But if you're asking for SARS-CoV-2, it's not an issue. But if you're looking for lateral flow for swine blood in general, I'm sure it will work.
All right. So this one's the last one from Preeti K. Patania, who asks, so she thanks you for the valuable information. What is the success rate of in silico predicted epitopes as vaccines, and how can we make them more suitable in terms of confirmation so that they can match as they are in native proteins?
That'll be another hour lecture for you, Preeti, but I will try to answer this in a short way. So I mean, the techniques today, they're growing, right? I mean, so we're learning more and more from the standpoint of whole genome sequencing and looking at the different things that we can annotate. The problem with whole genome sequencing is so much data. You need to be able to really look at and intelligently analyze what you have generated, right? So really, from a data scientist perspective, you need to have intelligent people to look at that volume of data to predict what those things are. But having said that, there are many ways, from peptide-based arrays to other ways, to expression systems, to at least to predict what we can have from a literature in silico way, from a genetic sequence. But then we always have to express that material and then really test it. You can test that multiple ways because we already have antibodies against the pathogen. You can cross neutralize to see what it is. But I think where people fail is they expect that one single epitope to do everything, right? But I think the secret really is identifying a cocktail of these epitopes and then using that in the right combination to give you as good an efficacy as the real thing, as a real bug. So I think we always have the real vaccine, a virus. You can use it as a control to check it out. And I think you'll always want to triangulate. You want to make sure you compare that with this and to see it. And remember, it's not one single bullet that's going to solve the problem. You may want to look at multiple epitopes to give you the best possible combination. It's one thing to have a diagnostic assay and an epitope-based prediction, another thing to have a vaccine. So vaccine, you need multiple antigens. I'll leave it at that.
Got it. Thank you so much. This is again a very valuable interaction. Thanks to the audience for their participation. We won't be able to take any more questions, but if there are more questions that you have, please definitely email them to us and we try to answer those questions back. I would now like to request Dr. Kundu to give concluding remarks and close today's session, please.
Thank you so much, Malvika. And for all the others whose questions were not answered, we are going to make a compilation of that, share it with Mahesh, and try and get an answer for you. I know he's extremely busy and we are very, very grateful for his time. So Mahesh, thank you so much for your time, effort. And, I think you've said it all. In all that is attitude, sincerity, and working hard. With that, thank you so much, Mahesh. Thank you to the audience. We've had a great crowd today. And thank you so much, Malvika. We bring our episode of Biotechnology to a close. We come again live on January 13th, where we bring you 3 companies, small companies across the globe, together on a panel session. So we hope to see you all then. Thank you so much. A very, very good night from all of us. Have a great day ahead to all the people across the world. Thank you. Bye-bye. Thank you, Mahesh. Thank you, Malvika. Bye.
Bye.
Thank you.